Hello everyone, welcome to today's section. Today we are going to discuss about the specimen preparation for biochemical investigation. Myself, Sri Lakshmi, 2nd MSc Biology from St. Mary's College, Thrissur. Specimen preparation. The specimen and slide preparation is a crucial step for the histological or cytological observation. It is essential to highlight what needs to observe and to immobilize the sample at a particular point in time and with characteristics close to those of living state. Mainly, there are 5 steps for the preparation of specimen. First one is fixation, second one is embedding, the third one is sectioning, fourth one is staining and immunolabeling, and the last one is mounting. Fixation. It is carried out immediately after the removal of sample to be observed. It is used to immobilize and preserve the sample permanently as lifelike as set as possible. It can be performed using a fixative solution or by freezing. Fixation in a fixative solution is carried out by immersing the biological material in a formalin solution. Fixation by freezing consists in immersing the sample in a tissue freezing medium, which is then cooled in liquid nitrogen. The frozen sample is preserved until the sectioning. This step is carried out in a mold according to the orientation selected for later sectioning. The second step is embedding. Embedding is the step that follows fixation in a fixative solution. It consists in hardening the sample in a paraffin embedding medium in order to be able to carry out the sectioning. It is necessary to dehydrate the sample beforehand by replacing the water molecules in the sample with ethanol. As ethanol and paraffin are not miscible, an intermediate clearing agent that is miscible with paraffin is first used. After clearing the step, the samples are plunged in paraffin bath. This step is carried out using a dehydration automaton. The following embedding step is performed by using an embedding center. The sample is placed in a mold fixed with molten paraffin and it is carefully oriented according to its plane of section. The resulting block is then cooled. The next step is the sample is sectioned using a microtome. The third step is sectioning. It is performed by using a microtomy. Sectioning is an important step for the preparation of slides as it ensures a proper observation of the sample by microscopy. Paraffin embedded samples are cut by cross section using a microtome into thin slices of 5 micrometer. Glass slides are covered with a solution that contain an additive in order to keep the section attached onto the slide. Slides are then placed onto a hot plate in order to ensure a uniform spreading of sample. Once the slides are heated, the residual liquid is removed by hand. The slides are then dried at room temperature. Frozen samples are cut using a cryostat. The frozen sections are then placed on a glass slide for storage at -80 degrees Celsius. The choice of these preparation conditions is crucial in order to minimize the artifacts. Paraffin embedding is favored for preserving tissues. Freezing is more suitable for preserving DNA and RNA and for the labeling of water-soluble elements or of those sensitive to fixation medium. The fourth step is staining and immunolabeling. Staining increases the contrast in order to recognize and differentiate the different components of biological material. The sample is first deparaffinized and rehydrated so that the polar dyes can impregnate the tissues. The different dyes can thus interact with the components to be stained according to their affinities. Once staining is completed, the slide is rinsed and rehydrated for the mounting step. The immunolabeling will detect the particular cell and tissue components by using polyclonal or monoclonal antibodies. A solution which contains the antibody is deposited onto the sample, which is then washed in order to remove the unbound antibodies. The last and final step of specimen preparation is mounting. Sections are mounted between slides and cover slip using a product to ensure adhesion. The slides are then ready for the storage or observation. For the fluorescence microscopic observation, a mounting medium is used in order to temporarily decrease the fluorescence loss. The difference of specimen preparation in scanning electron microscopy and transmission electron microscopy. For TEM, the sample is fixed, post-fixed, dehydrated and embedded in resin. The sample is then microsectioned and mounted onto a grid for TEM imaging. In the case of some, the sample is fixed, post-fixed in some cases, dehydrated and mounted onto a step. Finally, the sample is coated with carbon or gold to make it conductive for imaging. Thank you.